Good afternoon, ladies and gentlemen, and welcome to the second event in our Prize Winners Lecture Series from the Bayer Foundation. I'm really honored today to be able to introduce this year's winner of the Otto Bayer Award, Professor Ruth Ley from the Max Planck Institute for Developmental Biology. Developmental Biology in Tübingen. I think we've lost my presentation. I will just share. I'd like to begin today actually, first of all, by saying a big thank you to all the people that have helped today's event come to being. So in particular to Selby Anayeva and Lars Sumulin, who have really helped us put today's event on, and also to our events support team. Secondly, before I introduce Ruth in a bit more detail, I would like to give a short introduction to the Bayer Foundation. So my name is Karl Collins, and I'm the director of the scientific program within the Bayer Foundation, and that's one of two key programs. We have the science program and the social innovation program. And our vision is to catalyze advances in these two areas towards a world with health for all and hunger for none. And this is a vision that we share with Bayer as an organization, but we are a legally independent organization, and we are a foundation as you would commonly understand the term. More specifically, within the science program, we seek to enhance the impact and value of science as a basis for societal progression. And we have a number of different programs in three key areas. First of which is to identify and nurture innovative scientists and cutting-edge research. Of course, our awards are part of this. We offer fellowships to up-and-coming scientists, and in the next year, we are developing a program to identify sort of those exciting areas of research on the periphery of academia and bring these more into the limelight. We seek to engage and inform, so the scientific community, such as yourselves, but also society more broadly around science through a series of events. We support external activities such as the Human Force competition here in Germany, and we have a whole host of educational programs. Final pillar of our activities is focused on building capacity and equality globally for the scientists of today and tomorrow. And in the upcoming weeks, we would share a lot of exciting news in this area. Specifically, we hope to launch our first collaboration supporting specifically science and scientists within sub-Saharan Africa. We also have a collaboration together with the Falling Walls Foundation supporting young female talents in science. And finally, we support the Christiane Nusslein-Volhard Award, which is a very important initiative that supports academics with children, supports female academics with children. Underpinning all of these activities, you can see over the next years, we seek to build an active and engaged community or network. As what we see as the role of the Bayer Foundation sitting at the center of all of these activities is that we have the ability to bring people together, to facilitate interdisciplinary discussions, to build support networks, and to provide opportunity for cross-fertilization of ideas. Now, this community is a community we hope Bayer scientists will engage with in the future. And should you wish to be involved in any of our activities, please feel free to contact either the Bayer Foundation email address, so we will share these in the chat directly, or contact me directly. We already have a number of active participants, either acting in an advisory capacity as mentors for our scientists, or actually involved in individual projects. So please feel free to reach out. So now coming back to the topic of today, so the Bayer Foundation Awards have really an outstanding history in identifying really top talent. Today's presentation will be from this year's Otto Bayer Award winner, which is given for the life sciences. On the left-hand side, you can see a former prize winner, so Christiane Nusslein-Volhard, who was also since a Nobel laureate. As part of our awards program, we also have the Family Hansen Award, which is given for medical research. And again, two very visible faces on the left-hand side, former recipients of this award, Professor Stefan Hell and Professor Emmanuel Charpentier, who won the Nobel Prize last year for chemistry for her work in CRISPR-Cas. In addition, we have awards, the Biothrombosis Research Award. I think really excitingly for me, we have the Early Excellence in Science Awards, which are given in chemistry, biology, medicine, and data science. For the Bioscience Fellows, you will have received an invitation to nominate potential awardees in this category, and we're really happy to see you're already engaging, and we encourage you to keep nominating people. And for those of you who didn't receive a personal invitation, please reach out to the fellows or your leadership within your departments. We're really happy to receive nominations from across the organization. So finally, just to comment that the BioFoundation Awards, we will host 6 events throughout the year. This is the second event, and the next one will be in May, where we invite Marinka Zitnik, so the Early Excellence Science Award winner for data science. Well, with that, I'd like to take the opportunity to introduce today's speaker, so Professor Ruth Ley. So Ruth, please feel free to take over and share your slides. Professor Ley is currently the director of the Max Planck Institute for Developmental Biology in Tübingen. She began her career studying integrative biology at the University of California in Berkeley, before moving to the University of Colorado in Boulder to undertake her PhD in Ecology and Evolutionary Biology with Professor Stephen Schmidt. She then stayed at the University of Colorado to begin a postdoc career with Professor Norman Pace, studying molecular, cellular, and developmental biology. And I think it's at this stage in Ruth's career that she made the transition from studying, if you like, the soil or the microbiome associated with the plant world into the human gut microbiome. She joined the Washington School of Medicine at the Center of Genome Sciences with Professor Jeffrey Gordon. Following that, Ruth moved to Cornell as an assistant professor and was promoted to associate professor in 2013 before taking up a really illustrious position here in Germany at the Max Planck as director of the Department for Microbiome Science. So Ruth has won a number of awards and honors throughout her career, in addition to today's Otto Bayer Award. But I'd just like to highlight that since 2017, Ruth is a Thomson ISI highly cited researcher, which really indicates the relevance of her research in the current environment. And last year, she became a member of the Scientific Council of the Pasteur Institute, really a position of great honor. So with that, I would like to hand over. I would like to welcome Ruth to the floor to begin her talk. Say thank you very much for being with us here today, and we're very excited about your presentation.
Okay, thank you, Karl, for that really kind introduction. And oh dear, let's see, my slides are not moving. There we go. All right, and I'd like to thank the BioFoundation once again for the Otto Bayer Award. It was a lovely surprise and a really great honor. So thanks very much for the award and also for inviting me to speak to you today. So as Karl mentioned, my independent research career started at Cornell, where we were for 8 years. And we recently moved, well, not so recently anymore, but 5 years ago, we made the transition to the Max Planck for Developmental Biology, where Christian Nüsslein-Foltard actually is in Tübingen. And I want to begin by thanking the members of the lab shown here through various generations, because the work that I'll be showing you today was done by them. And I made my title deliberately vague so that I could sort of pick and choose amongst some of the work that these guys have done that might be of interest to you. We work on the microbiome of the gut, and when I use the word microbiome, I really mean the community of microbes in the gut, the assemblage of microbes. I use that quite loosely as meaning just the collection of microbes that are there. And so I'm going to divide my talk into 2 parts. The first part, I'll tell you about what we've found out about how composition of the microbiome relates to host genetic variation, and how we followed up on some of the findings there. And in the second part, because I think there's chemists in the audience who might want to see something, or at least Karl is, see something a little more molecular. I'll talk to you about our recent work that we've started to look at bacterial sphingolipids and how they might affect the host lipidome. So the human microbiome, as you've probably heard by now, is made up of more cells than it's made up of a huge amount of cells, right? But they add up to about a kilo or so in the gut, and they're mostly bacteria. They can also comprise some archaea and some other eukaryotes, and they're made up of hundreds, if not thousands, of species. Definitely a huge variety at the strain level, and they encode a very large genomic capacity that really can extend our own phenotype. We focus on the gut, which is about 40 square meters. And here, especially in the distal gut, this is where microbes enable food digestion. They're part of our nutrition. And then there's a lot of interactions with the immune system, which are particularly interesting for a variety of disease development and so on. They also play roles in infection resistance, detoxification, and in the second part of my talk, I'll mention how they can, we think, provision us with special lipids. We know from previous work that the microbiome can impact our phenotypes, particularly host metabolism is one that they seem to have a particular effect on, through effects on insulin resistance and the development of metabolic syndrome and diabetes. And the causal link between gut microbiomes and these metabolic phenotypes was really established through these kinds of experiments where microbiomes were taken from lean or obese donor animals at first and transplanted into germ-free mice. And so these are raised aseptically without any microbes at all. They're empty vessels, if you will, kept in aseptic isolators. And you can transfer a microbiome into one of these mice and then look to see at how the phenotype changes. And the obese donor microbiomes often, or consistently, I should say, transfer some of those metabolic phenotypes. And so this was originally shown with genetically obese mice, OB mice, then mice that were obese due to their diets, germ-free mice that were given human microbes and made fat due to their diets. And then finally, with human lean and obese donors, this also transfers some of this phenotype as well. So here are some of the papers listed that I was involved in with these kinds of experiments, but this is something that's really very robustly replicated across laboratories and very consistent. So there's a lot of interest, of course, in the mechanisms underlying this and how this works. And there's various ways this happens, namely metabolic inflammation. So to backtrack and sort of ask, where do these microbes stem from? When we're born germ-free, and we're rapidly colonized at birth by microbes from our family members, our mother in particular, our caregiver, primary caregivers, family members, quite possibly pets. And of course, children are also exposed to environmental microbes from food, from soil and whatnot. But these tend to be transitory, and these don't tend to take up residence in the gut. So as kids grow up, as people grow up, there's increase in complexity and diversity and biomass, so that by the time you're an adult, you have somewhere in the order of 10 to 100 trillion microbial cells and a unique signature to your microbes, because you've had a unique history of microbial colonization and so on. And so there's been a lot of work into the various environmental factors that affect not just the seeding, what you're seeded with essentially, but also the composition, the relative abundance of these different types, and how that relates to diet, health, lifestyle, and so on. And in my lab, a question that we asked is, what about the genetic makeup of the host? You know, how might that influence the composition of the microbial community that you have in your gut? So another way of posing this question is, do people have a particular genetic predisposition for a certain kind of microbial community composition? Do you have a genetic predisposition for a lot of microbe A or microbe B compared to somebody else that we can relate to your genotype? And when we started this in 2008, the answer was no, or the dogma on the field at the time, which was very new, was no. That studies that had looked, or really this was the big study at the time from Jeff Gordon's lab, had asked, do we see an effective genotype on the overall diversity of the gut? And the answer, based on comparing different kinds of twins, was no. But that was based on comparing overall diversity between people and only 54 samples, and we felt at the time that this was underpowered and that there could be more there. So we undertook, with the help of Tim Spector at King's College London, who runs a large twin registry in the UK, we undertook a study of the microbiome of twins. So we looked at over 1,000 twin pairs who are either fraternal or identical, and we sampled all these English people for their gut microbiomes. And the advantage of working with this twin population was that they were heavily genotyped and phenotyped at the time. And so I'm going to refer back to this paper that came out in 2014 because it's laid some of the foundation for the work that I'll tell you about that we've followed up on since then. But one of the main questions, and the reason that we went with twins, is they're very handy for asking this kind of question: are any of the gut microbes heritable? And this means, to what extent do genetic factors influence the differences between people when we look at their microbiomes? And so the same way height is influenced by your genes, can we apply that to microbial traits and ask, if everyone's got E. coli, can we say that there's a genetic predisposition for having more or less of E. coli, for instance, and then apply that to all the gut microbes and ask which ones do we see a genetic influence for? And so the way we do this is by comparing twins within twinships, and whether they're monozygotic and dizygotic. So you can do this for any quantitative trait, and in our case it would be abundances of microbes. Monozygotic twins have the same genomes, and dizygotic twins only share 50% of their genes, and so you can model these kinds of data to get a strength of the genetic effect, to ask just how much of a genetic effect there is on the composition of, on the relative abundance of the different microbes. So we did this for over 1,000 twin pairs, and this figure boils the data down to a single figure. So what you're looking at is a phylogeny of all the microbes that we found in these people, and we're asking about the strength of this genetic effect, or the heritability, at each and every branch. And if there is an appreciable genetic effect or heritability, you're going to see a warm color. So you can see right away that there's a lot of blue and green, meaning for the majority here, for 90%, there isn't a genetic effect on that we can pick up on the variance in the abundances of these microbes across these people. But you can see also, you can see some red branches peeking out, and these are our so-called heritable microbes. These are the ones that we are genetically predisposed to have more or less of them. And so we're zooming in on these and asking what they are. And this is a list of them down here. And what this now is showing is that other studies have since corroborated this, and from different parts around the world, you can see, from different studies around the world, you can see that the same heritable microbes are showing up again and again. And so now we have a list, and we can ask, why are they heritable? What aspect of post-genetics is driving this heritability? And do they have a particularly special relationship with their human hosts compared to the ones that aren't heritable, for instance? I'm going to tell you about a couple of these, the Christensiaceae and the methanogens. And the reason I want to tell you about these, first, the family Christensiaceae is the top most heritable one that we found, and that's why it's in red in this network. What this network is showing you is that it co-occurs across the twins with these other families that are clustered with it in this co-occurrence network. And so a lot of these have kind of weird names, and that reflects the fact that there's no cultured representatives for these. But there is for the Methanobacteriaceae, and that is really coming from the dominant methanogen in the human gut, which is Methanobrevibacter smithii. So we've got a co-occurrence of our most heritable set of microbes here in this family, and the dominant methanogen in the human gut. So since then, we've looked to see if this was something unique to English twins, or if we could see this more broadly. So this is an analysis we did of a meta-analysis of 10 studies that included about 2,000 shotgun metagenomes. And so in this case, people have gone out and shotgun sequenced microbiomes, and that way you can get an idea of the genomes of the microbes, not just marker genes. And what we find here for these then is that across these studies, and you can see where they came from around the world, we see a nice correlation between the methanogens and the Christensiaceae again. So this is something that isn't just English. We pick it up in virtually every study we look at. So there's something, these two, they either like each other a lot for some reason, or they like the same conditions. What we also noted, and this was a surprise at the time, because my early work was on obesity, and I didn't expect a study on host genetics to bring me back to it. But what we found was that this whole co-occurrence network, this cluster here, was enriched in lean versus obese people in the study. So there's an association of these guys with leanness, meaning lean people have more of these on average. And when we looked across these 10 studies, we picked this up again. So across these 10 studies and about 2,000 metagenomes, we saw that not only are these two types of bacteria, these families, correlated with each other, but they're highly prevalent. So you can see that they're in like pretty much everybody, and they're also correlated with a low BMI across populations. So these, we had picked this particular set of 10 studies due to the fact that age, BMI, and other things that we can correct for were reported and available to look at. So we've kept an eye on the literature since we reported an association in 2014 of the Christensiaceae, in particular with a low BMI. As of the beginning of the year, this is the number of studies, and you can see where they're coming from and the size of the study. So something quite big. This is a number of people in each of these studies reporting an association between the Christensiaceae and a low BMI. So it really is emerging, I think, as a marker for a healthy body weight over and over again. Not only BMI, but also if we add in studies reporting the association of these bacteria with a healthy metabolic state, such as healthy levels of triglycerides, for instance, we can really extend that and see even more studies, more parts of the world of reporting associations. And so this again sort of indicates that there might be something interesting going on with these microbes, and it wasn't just something that peculiar to English people, if you like, but that we see around the world. So to get back to these two microbes that we could work with, because just as we published this particular study, that type strain of this family had just become available. These are also available to work with in the laboratory, not so much the others because they're still uncultured. So what are they? I'll just tell you a little bit more about them. The type strain of the Christensiaceae family is Christensenella minuta, and it's a relatively small family right now within the firmicutes, so the gram-positive firmicutes, which make up, firmicutes themselves are a dominant phylum in the human gut. These have a few cultured isolates, and there's quite a bit of uncultured diversity that we know from putting together genomes from metagenomes, for instance. But from looking at them and their genomes and working with them in the lab, they're non-motile, fermenting bacteria in the gut. They don't have a very large repertoire of carbohydrate active genes like the Bacteroides might, so they look like they're taking relatively simple sugars and fermenting them. On the other hand, the Methanobacter smithii is an archaeon. It's the most prominent archaeon in the human gut, and what it does is take the products of fermentation, hydrogen and CO2, and make methane. So you can see that there's a reason for these to be together, is that there could be cross-feeding here. So we looked to see if there was indeed cross-feeding. So this is what Christensenella minuta looks like by microscopy. It makes these really nice rosettes. It actually sticks together a lot. It forms biofilms and clumps readily. And this is what Methanobacter smithii looks like. It has to be grown usually under pressurized hydrogen if it's grown alone. You can make Methanobacter smithii very happy by tossing it in with C. minuta, and it will go right into the biofilm. So here again are these rosettes of C. minuta, and here you've got the fatter cells of M. smithii. So there's fewer of them, but they contact directly the C. minuta cells when grown together. And in fact, they do much better together than they do alone. So here they're grown under continuous flow in bioreactors in the lab, and here by qPCR we're just measuring how much biomass we get. And when they're grown together, they consume more glucose in the system, or C. minuta consumes more glucose, and both do better than when they're grown alone. So in this case, this would be grown under excess hydrogen and CO2, the smithii, but smithii does better when it's with C. minuta than when it's alone, and they both do better. So they seem to definitely be in some kind of cross-feeding syntrophy here. Indeed, if you toss them in a bottle, what you'll see is that hydrogen levels initially go up as C. minuta starts to ferment substrate, and those hydrogen levels will be drawn down along with the production of methane. And you can see this is another visualization of those methanogen cells nicely in with the with C. minuta. A question that we had about C. minuta and how it was associated with the phenotype is, could we actually, now that we know that there's some sort of genetic influence on the abundance of Christensenella, could we actually add it to an obese microbiome and see an alteration of phenotype? Of course, not this drastic. But can we do something like this? And so what we chose to do was to take a microbiome from an obese donor that essentially lacked Christensenella and either add it dead or alive, mix it in, if you will, dead or alive cells with a donor microbiome, give that to germ-free animals, and we would expect that those receiving the live Christensenella wouldn't develop as much adipose tissue. And that's in fact what we see. Here are some data that are unpublished but representative in male and female mice, 16 here per sex, and these we've repeated this a number of times. You can see that overall there's less weight gain and there's less total adiposity on the animals if they receive live versus heat-killed Christensenella. So in a mouse model, we can replicate this this phenotype effect, if you will, by adding Christensenella. The paper just came out last week, actually, by a company, Ecology Bioscience, that has a strain of Christensenella minuta that they are now giving to, that they first checked out in diet-induced obese mice. So I should say the previous experiment that we did, we have the mice on regular chow, regular mouse chow, polysaccharide rich. Here they're diet-induced obese, and the green is their addition of their C. minuta strain, and they're reporting less weight gain and better cardiometabolic markers when animals receive the live Christensenella strain. And it's a new strain because they're developing this to give to people, which would be pretty interesting, and they've actually just registered a clinical trial, a first safety trial of giving people this probiotic. So that's going to be interesting to keep an eye on. And we'll see. Well, I'm not involved, but it's interesting to see that this is being taken further to see how it might work in people. we're basic researchers and still interested in this genetic question. And so what we've been able to do with the twins, because they were genotyped, is ask about these heritable microbes. What are the genes in the human genome that might be implicated in driving the heritability of these microbes? Can we actually start to see on the human genetic side what's involved? I should tell you right now, there's nothing for Christensenella or the methanogens, so I'm going to tell you what there is evidence for. This is like the strongest signal to come out of this kind of study, and it relates to the genotype that's predictive of whether or not people are lactase persistent or not. So there's people with this non-lactase persistent genotype that are unable to metabolize the milk sugar lactose, and those people have higher levels of bifidobacteria compared to people who continue to make lactase into adulthood and can metabolize themselves the milk sugar lactose. They have lower levels of bifidobacterium in the gut. So there's a very strong association here between levels of bifidobacteria, which are known as lactose consumers, with the human genotype that tells you whether or not you're lactase persistent or not, and that relates pretty well with lactose tolerance or intolerance. And lactase persistence is something that evolved in a couple different places around the world independently during the Neolithic transition, particularly Northern Europe, but also parts of Africa and Asia independently saw humans develop lactase persistence into adulthood due to the fact that they were domesticating animals and drinking their milk. And so that got us thinking about whether heritable microbes were reflecting some of these large genetic differences that we see in human populations, as the human geneticists report, for the genetic variation that we see around the world when people moved into new environments or changed their diets and adapted to new conditions. And so there's this existing variability in between people that relates to some of these adaptations, and we can perhaps start looking there for some of these microbiome effects. So I've just told you that we see that very strongly with the milk adaptation. We've also looked at starchy food adaptations, and that has to do with the copy number of the salivary amylase gene. Starches begin to be digested in the mouth through salivary amylase, and the more salivary amylase you have, the higher your copy number for that gene. It's a very simple, beautiful relationship. There's been duplication of the AMY1 gene locus, and people with many copies, it can go up to 20 copies, have much more of the enzyme amylase in their mouth, and they begin they're going to be pumping more of this enzyme into the system, if you will, so that if you have high levels of the amylase, your own enzyme will be accessing some starches from your food and altering what your microbes see. Whereas if you have low copy numbers, the microbes are going to be and low activity, the microbes in the gut are going to be seeing something a little different. So we've been able to compare people with high and low copy numbers and find that ruminococcus, which are known to access resistant starch, are very responsive to whether or not you're high or low on this axis. And so that's another example of human genetic adaptations to diet during the Neolithic, that we carry the variation with us today in our populations, and we can actually see that the microbiomes respond to that in present-day populations. And it's going to be interesting to see how that actually relates to our health now that we're eating different kinds of food from when these adaptations occurred a couple tens of thousands of years ago, for instance. So going back to what the human geneticists have shown us, that there's this map now of human genetic adaptations to conditions around the world, from not just diet, but also high altitude, pathogen pressures, and so on. If we look through the literature, researchers have associated all of these different genetic states in the human populations with differences in microbiomes here. So wherever there is one of these genetic differences, you could typically find a microbiome difference that might be reflecting that. And so Taiichi Suzuki in the lab, for instance, we recently summarized our thinking about that. If you'd like to read more about it, we've started to think about what this really means for our interaction with the environment being filtered through the microbiome, and how has the microbiome helped or hindered, if you will, human adaptation to novel environments. So if you're interested in reading more about that, we've summarized what we've been able to glean just recently this year. So I'm going to switch now to something a little more molecular, and talk to you about what we've been doing recently with sphingolipids in the hosts and produced by bacteria, and how they might affect host lipidomics. So if you go back to thinking about what microbes are actually doing for us, they help with digestion of foodstuffs. They provide us with extra nutrition in the form, for instance, of short-chain fatty acids, that being butyrate, acetate, propionate, and so on. And butyrate being, for instance, the primary food source for the colon cells. So 80% of the energy that the colon uses comes from fermentation of fiber by the gut microbes in the form of butyrate, and it's also an important anti-inflammatory. There's a lot known now about immune priming of microbes, particularly in early life, and how that affects health states later on, and allergy and allergic disease, for instance. They can be very important for resisting pathogen resistance, for resisting pathogen infection, and so forth. And we've been starting to ask what other kinds of molecules can mediate these interactions. And so we often talk about the products of fermentation, for instance, these short-chain fatty acids, and we often talk about the various types of molecules that interact with the immune system, such as flagellin or LPS. But we started wondering about lipids themselves that make up the microbial membranes. And this is because of a particular characteristic of the Bacteroidetes that caught our interest. So the phylum Bacteroidetes makes up, on average, about half of the cells in the human gut. This is illustrated here in this bar graph. This is about 3,000 samples, and you're looking at the frequency of different types of bacteria, and they're colored in red here is the Bacteroidetes, and they're ranked by abundance. So you can see that some people have very high, some people have very low. On average, you're at about 40%, and this is pretty typical, not just for Western populations, but also non-Western populations. And if Western and non-Western populations can differ by the specific taxa that make up the Bacteroidetes, but they're still Bacteroidetes. So for instance, if you look at the American Human Microbiome Project data, the twins from the UK, other US data sets, you see a lot of Bacteroides, which is a large genus within the phylum. If you look at people from Africa and South America, you'll see more Prevotella, but that's a sister taxon within the within the phylum. So it's one or the other, but regardless, they share this property of making sphingolipids. So here are this is a big tree of our latest understanding of how the deep phylogeny of these different bacterial lineages and some of the archaea. And here are the Bacteroidetes. So they make sphingolipids, and there's only one other, oops, well, my arrow went away. There's only, okay, here we go. So there's one other group of bacteria that make sphingolipids, and those are some of the alpha proteobacteria, things like Sphingomonas, and these things are associated with plants, and often, so you find them often in plant leaves, for instance. So you've got bacteria that are in the mammalian gut. So the Bacteroidetes are not just in the human gut, but they're in mammals and vertebrates in general, and very adapted to them. So they're making sphingolipids, and then these things that live in plant leaves are also making sphingolipids, and that was something that seemed a bit coincidental, and it got us to wondering what was going on with sphingolipids. So they have sphingolipids in their membranes. The word sphingo comes from sphinx, but really they're one of the 8 major classes of lipids, and there's, like all different kinds of lipid classes, they can have an immense variety because you can switch out different parts. And so this is an example sphingolipid. They all have a long chain base, and then they have this fatty acid chain, and then you can switch out these head groups. And depending what the head groups are, they can have very different roles. And these roles have to do with not only membrane composition, but also signaling. So there's various diseases that are related to abnormal sphingolipid metabolism, such as Tay-Sachs, and some of these are hereditary. Also recently, in the last 10 years or so, there's more and more evidence that when ceramides accumulate, that is linked to metabolic diseases such as obesity and diabetes, for instance. So they're very important for human biology in a number of different ways, and there's a huge variety of these across the body doing different things. Eukaryotes, all eukaryotes make sphingolipids, and this is the first step in the synthesis with the enzyme SPT that brings together palmitic acid and serine in this first synthesis of sphingonine, which can then go through various transformations into ceramides, and ceramides can then be transformed into other things such as sphingosine 1P. But this is the very first step, and it goes from there into, for instance, also into much more complex sphingolipids, such as the ones in the brain, depending then what the head groups are and so on. If you look at the mammalian sphingolipids compared to the bacterial sphingolipids, they're quite similar in structure. One of the important differences between the mammalian and the bacterial sphingolipids is that the mammalian's sphingolipids have these even length chains, whereas the bacterial sphingolipids can also have odd chain sphingolipids, and this makes it handy for trying to track some of these by mass spec, for instance, because only the bacteria will make these odd chain ones. So along with Liz Johnson, who's now at Cornell, professor at Cornell, we started to ask in human biology, what are the sources of these lipids? And so one important source is de novo synthesis, but it makes up a small proportion of the pool in the body. It's a very important pool. It's actually, if it's knocked out completely, you can't knock out SPT in a mouse, for instance. That's lethal. So it's very important to have de novo synthesis, but the bulk of the sphingolipids do come from the diet. The diet, of course, has to have sphingolipids in it, and the mammalian or animal-derived sphingolipids are much easier to access through digestion than plant sphingolipids, for instance. But as I told you, you're carrying around in your gut a substantial mass of gut bacteria that are also making sphingolipids, and so we wanted to ask if they could contribute to these pools, particularly ceramide pools, which I told you have been linked to metabolic disease. So if we look, and these are samples taken from the twins again, we pooled people that were high on the Bacteroides scale or low on the Bacteroides scale. This is looking at the makeup of the gut bacteria. We can find people that have at least 50% and some that have very little. And if we look by mass spec at their different sphingolipids, we can see for the even chain and the odd chain sphingolipids, people with the high Bacteroidetes, or have, in fact, high Bacteroides in this case, have higher levels of these sphingolipids in the gut, which makes sense. So we can see just by looking at the gut contents that there's more sphingolipids when you have sphingolipid-producing bacteria present. Stacy Heaver, who's a graduate student in the lab, started working with Bacteroides theta iota omicron as a model system. So this is probably the most famous Bacteroides that has been worked with in labs across the world, Fragilis being another, for instance. But this is a representative commensal beneficial microbe, prevalent in the human gut, makes sphingolipids. And the nice thing about it is that there's genetic tools, and so we can work with it. One of the things we did with it was to ask whether we can see lipids from B theta, I'm going to refer to it as B theta, moving into the animal host. So here what we did was grow B theta with alkyne labeled palmitic acid. So this is click labeling, put those into germ-free mice and then ask if the label moved. And in fact it does. So this is germ-free animal, this is the gut sections of germ-free animals. This is small intestine. This is our click-labeled bacteria added, and after a few days, you can see the alkyne-labeled lipids have now moved into the epithelium. So this could include sphingolipids. It probably includes others as well because we put the click on the palmitic acid, but it does show that we can see lipids that were originally in the bacterial cells moving into the host tissue. What Stacy did was take B theta and knock out SPT, and surprisingly, the bacteria tolerate this. So we can take a sphingolipid producing Bacteroides and we can eliminate its ability to make sphingolipids. So this is showing you the knockout delta SPT. It looks about the same. It's got maybe some different outer membrane or capsule properties, but it's viable and we can work with it. These types of bacteria make a lot of outer membrane vesicles, and this is one way we think that the lipids and other components from bacterial cells can move across the mucosal layer and be delivered to the host epithelial cells. So if we look in their OMV fractions shown here for both B theta and the SPT mutants, they still make, we can pick up the OMVs, and there's definitely no sphingolipids in the delta SPT OMVs, but there are plenty of sphingolipids, just like the cellular fraction for B theta. What Stacy's also done is put SPT under an inducible promoter, and this has been a lot of fun for looking at how the knockout responds to having SPT removed, and then if we slowly dial it back in, how the membrane responds. And I'd be happy to show anyone who's interested more of that separately, because we've looked quite carefully at how it responds to tuning it back up and down. But that's I think if you're interested in that, contact me. What we can do with B theta and the delta SPT is colonize them in germ-free mice, and they colonize the same level. So that's interesting, and it allows us now to compare directly with the same kind of microbe what happens when it does or doesn't have the capacity to produce these sphingolipids. What's the effect on the host? So here are these kinds of experiments where we've taken germ-free animals and we've colonized them with either B theta or the knockout for the SPT, and we can compare them to germ-free. And we're now looking at sphingolipid levels in the ceca, for instance. You could see that they're much, much higher with B theta present, and of course lower with the delta SPT. So you can see definitely this is what you'd expect. And then when we start looking in tissue, we can also see in the ileum tissue that with B theta present, you have more of these sphingoid bases that you can pick up. In the ileum, we pick up higher levels of ceramides as well. And then we can move into the liver and see that we also have higher levels of sphingolipids in the livers of mice that are colonized with these bacteria that can make sphingolipids. So here are the ceramides, the different kinds of ceramides, and pretty consistently there's higher levels of sphingolipids when the animals have B theta, which produces sphingolipids. So in this particular system, you can definitely see that having sphingolipid producing bacteria in the gut is altering the liver ceramide levels. Here we took not germ-free animals, but conventionally raised animals and placed them on a fatty acid-free diet. And what this does is force the animal to ramp up that de novo production. So they're having to work harder to make sphingolipids in this case. So this would be the de novo production. We're going to be looking at rates of de novo production for the ceramides. And so for control animals, or for animals that we're administering the bacteria to them, they have their own microbiomes, but we're amending them with extra bacteria by oral gavage. And if we give them the delta SPT bacteria, we see no difference compared to control with the rates of de novo production in the liver. But when the mice get B theta, which has the lipids, the sphingolipids, we see that the animals basically have to work less, right? So it really is, even in a conventionally raised animal with a normal microbiome, this is acting as a source of sphingolipids that the animal can use, and it doesn't have to work as hard to make its own sphingolipids. Here we had a diet-induced obese model of insulin resistance. So these animals are conventionally raised, normal microbiome, and they were fed on a high-fat diet. And then we administered them either B theta or the knockout to check what this would do to their liver ceramide levels. So they were kept for about a month on the high-fat diet, and then they were administered these daily doses of B theta or the delta SPT. And in these animals as well, giving them B theta led to higher levels of these ceramides and overall sphingolipids, as you can see from the sphingoid bases. What we also saw was a significant, small effect, but significant effect of insulin sensitivity, and this is consistent with what other people have shown when there's been studies that have altered levels of some of the ceramides in the liver, either through drugs or through genetic means, and seen that when you alter levels of some of these ceramides, you can see effects on insulin sensitivity. And so we're also seeing this just by administering these bacteria to the gut. So to sum up what we've been seeing so far, bacterial sphingolipids are produced in the gut. They can enter host metabolic pathways. They can change the levels that we see in the liver. And we think that they have the capacity perhaps to affect insulin sensitivity as well. So this has implications, I think, possibly in the human context, although this needs to be looked at directly. For what it means to be high or low on that bacteroides scale, you're going to be receiving more or less of these lipids possibly from your gut bacteria. So we've looked in the liver. We're now starting to look in the brain at the lipidome there, and this was motivated by thinking about what it means, especially for children. If their diet is very poor in animal products, and sphingolipids have to be coming from a plant source alone, which is much less available, do the bacteroides really become important sources of sphingolipids in those kinds of conditions, and would we see any implications in the brain and in brain development to be chronically undersupplied with sphingolipids, and can bacteria compensate for that? Is what so that's the bigger question. So to start looking at this in an animal model, we've been playing the same kind of games where we take germ-free animals, we give them B theta or the SPT knockout, and then by mass spec imaging, we're now scanning the brains of these animals to ask in what parts of the brain and for what gangliosides, for instance, might we see this being what's coming from the gut to be important or not. So that's where we're going with this. This is some of our first data coming off our new Bruker SciMax that we're quite excited to have. So getting back to what bacteria do for us, in terms of provisioning us with different types of molecules like these short-chain fatty acids that are involved in energy, I think we can also add sphingolipids to this. And I think it opens up a lot of questions about because there's so much complexity and diversity in the lipids, not just sphingolipids, not just those of Bacteroides, but there's a huge amount of really unknown diversity in the bacterial lipid field, and asking how this might be interacting with the host. I think we're at the very beginning of this, and it's going to be an interesting field to delve into. So I've told you about how some of the host genes can modulate components of the microbiome, how they might actually feed back on our own phenotypes. In my own group, we're trying to also not just look at these broader scale patterns, but follow some of the molecules to understand how this might be happening. And with that, I'd like to thank the people who supported the work, the Bayer Foundation again for this opportunity and the lovely prize. In the United States, this was all initiated through foundations as well, such as the David and Lucille Packard Foundation, the Beckman Foundation. So foundations are really terrific, especially I think for early career researchers, in giving you that extra boost and allowing you to take risks that some of the more conservative funding bodies like the NIH follow up on later. And now, of course, we have the Max Planck Society funding our work, which is allowing us a lot of freedom. So our partners include Tim Spector from King's College, Andy Clark, who's a geneticist at Cornell, Lars Angenant, who's an environmental engineer, Andrew Bewertz, who works on metabolism. And then at Tübingen, we've been very fortunate to work with Peter Kremsner and his associates in starting to ask these questions in other parts of the world, which I didn't get into, but we're starting to look outside of Western gut microbiome interactions, but looking at people from Africa and Asia as well, so to understand what we can generalize on and what we can't. So with that, I'd like to thank you very much.
Thank you very much, Ruth, for a really great and engaging presentation. I appreciate very much as a chemist seeing the occasion on chemical structure in there. That's really fantastic. And we had a huge amount of interest online with Q&A. So we don't have too much time, but I think we can go through the questions. We still have a few hundred people in the call, so we're peaked at around 300. And for those that are willing to stay, we definitely go through the questions. Okay. So the most highly ranked question came very early, but what is your opinion on microbiome testing for consumers? It's becoming more available and affordable, but is this an opportunity for awareness, or does it lead to overdiagnosing?
Well, that's an interesting question. I think if one is curious, it's a fun thing to do. If a physician is given the information on a patient, I'm not sure it's actionable at this point. So I see it from my perspective as something that's interesting for curious people to look at, which one should proceed with caution on how to and not necessarily act on it. Although there are companies now, such as Day 2, that are claiming that they can advise you what to eat based on your microbiome composition, and I think that may or may not work. I'm not entirely sure. But, so for me right now, I think it's early days to know how one might actually proceed with information like that. I think if your gut microbiome was extremely odd, that you would have other symptoms that might have led you to seek help in the first place.
I think related to what you said there's sort of external influences on the composition of the microbiome. We actually had a lot of questions, so asking about the extent the microbiome is influenced by diet and what is the long-term influence of sort of a current unhealthy diet on the microbiome. Also thrown into the mix there was the impact of antibiotic use, especially in early childhood on microbiome composition.
Right. So on a day-to-day basis, changes in the diet will modulate the relative abundances of the different taxa as they as they respond to the food that's coming through. And that's something that if you sample people repeatedly over time, you can see this up and down. That being said, if you switch your diet more and more, so if you start with a very unhealthy diet, Western diet, and you move towards maybe a Mediterranean diet, you're definitely going to see a shift that's more permanent with an overall shift, and it's very likely that kind of shift to a fiber-rich diet that's rich in fruits and vegetables and that kind of thing will also bring a shift in the proportion of the bacteria that's towards these more healthy ones that produce butyrate and things like that. Antibiotics, it really depends on which one you're taking and how long you're taking it. There's quite a bit of antibiotic resistance in the microbiome, so even one that seems to hit them pretty hard, you can often see that there'll be an overall reduction in the mass, but they'll come back. The thing is, they don't always come back the exact same way. There might be a slight rearrangement, for instance. What is more perturbing is if you take strong, antibiotics that are very strong against the microbiome repeatedly, again and again, you might start to see diversity drop, as you might start to lose things that don't come back. And this is what is concerning at a population level with Western societies in general. But combined with other things such as low fiber in the diet, low transmission rates between people, other factors, this is all combining to reduce the diversity that we're losing things. And similarly, this might be particularly important for kids. Kids are now, especially in the US, being administered courses of antibiotics much more frequently than in the past. And there is concern that this can alter perhaps what a healthy development might be, and that has consequences for the education of the immune system and educating the immune system to recognize self versus non-self, and that this might have something to do with development of allergy and other autoimmune diseases, for instance. So there's a lot of work into that.
So I guess, this ties again nicely into the next question, and I think you've kind of answered it. Yes, is there a measure of a healthy microbiome in terms of composition or characteristics? So I guess the diversity of the microbiome here is what you're alluding to. And the second part of that question is, what is your view on proxies for microbiome health, like pH or redox status?
Okay, so in terms of what a healthy microbiome is, I think that's really, really context specific. So this was illustrated to me by what we saw when we looked at pregnant women, for instance. So the third trimester microbiomes for pregnant women looked unhealthy, and they might have been if they'd been taken, but they're not within that context. So microbiome is extremely context dependent, I think, and in terms of whether or not it's going to be health promoting or not, which is also a difficult kind of concept. So I think we have a sort of a general idea of what microbiomes generally look like, and we call that healthy. And then you can look at really sick people, and if they're really deviant, you call that unhealthy. But I think that it's a fuzzy concept, the healthy microbiome, and it's very context dependent. And a proxy, you know, the thing is the microbiomes are healthy often when the people are healthy too. So whatever proxy for health is, you know, that's probably going to work for the microbiome too, I would think. They tend to mirror a lot what's going on in the host, and the question is always, do they contribute? Yeah. Brilliant.
So then we move sort of more into the connection, sort of coming to the genetic component of your talk. So taking it one step further, do the mice examples, obese, non-obese, sorry, did you do any of the research in the context of the microbiome and epigenetics, sort of moving beyond just the genetic aspect?
We haven't, but I think those kinds of studies are ongoing and of great interest. Right. I think just keep an eye out for those. They'll be coming.
Okay, and then a whole sea of questions. I think you might expect the link between the microbiome and different diseases, pathologies, specifically some in relation to inflammatory bowel disease, obesity, diabetes, but maybe you could just comment on this more generally.
Yeah, more generally. So, I mean, I think in the context of metabolic disease, we're looking at whether microbes can contribute to metabolic inflammation. So through interactions with the immune system, put the host in a state of low-grade inflammation that can then have metabolic consequences, and that can be triggered in animal models at least, and it's starting to look like in humans as well. When you put people on very controlled diets that include things like emulsifiers or very high sugar, the microbes that respond to that tend to be inflammatory, and they will have an effect on the immune system, and that can sort of get you in a feedback loop where this low-grade inflammation can be then maintained by the immune system. So that's one way, at least, that it looks like the metabolic inflammation can develop. It can be driven by changes in microbiota that come from diet. Other diseases such as Crohn's, colitis, that's a bit tricky because they're really autoimmune diseases, and it looks like the microbes are being possibly involved in driving some of that. But I think the underlying factors are often genetic to begin with, and the microbes provide a source of an additional source of inflammation, and it's also because of barrier breakdown, right? It's because they're no longer where they should be, and they're contributing things like flagellin and LPS that's driving some of that inflammation. Does that answer that?
Yeah, I think so. So must say we've come to the end of today. I want to apologize. We couldn't get through all of the questions. There really was a whole host. I think that gives you... No, not at all. I think it just is an indication of how engaged and how interesting people were in the presentation today. I want to finish by saying thank you very much to everyone for participating today. So more than 300 people on the call, which is really, really fantastic to see. And we look forward to seeing you at the next event in the Bio Foundation Prize Winners Lecture Series, and we will be in touch in the near future with the details.
Thanks so much. Please email me if you want to be in touch about any of this stuff, and especially chemists. I'm happy to keep the conversation going. So thanks very much, Karl.
Thank you so much for engaging with us again. It was really great to have you here. And if you could just stay on the call for a moment afterwards, that'd be fantastic.
Okay.
Thank you to everybody, and I wish you all a really great day.
Thank you.